Today I'll be doing my Unit 10 presentation, which is on drugs and medicine, focusing on the structure of drug molecules, isomers, their effects on a drug, and the methods by which drugs are detected. So let's begin with some definitions, and the first definition we're going to be talking about is the definition of a drug. And a drug is a chemical compound that interacts with the body's systems to produce a specific physiological response, and this includes effects that could be both beneficial and harmful, unfortunately. And the next definition is medicine, whereby a common medicine used every day is paracetamol, and it's used for things like headaches, pains, and general aches. And it brings me to the point that all medicines are drugs, but not all drugs are medicines. And regarding placebos, they're essentially the patient's belief that the treatment will be effective. And it's based upon the brain's response to expectations surrounding treatment. And a scenario in which a placebo might be used is in an experiment where one might administer a third of patients the placebo, a third of patients the drug, and the last third of patients neither the drug nor the placebo, and this is so that the people running the experiment can form a comparison point for new treatments, so that the medication being tested can be deemed safe and effective. And the next definition we'll be talking about is a therapeutic window, which is the range in which doses of a drug or treatment produce an optimal clinical response. And so drugs with a large therapeutic window have a better safety margin, which is very important. And the next definition is tolerance, and an example of this is when morphine is used for a long time, meaning larger and larger doses must be taken to produce the same effect. And this could happen due to the metabolism of the medication speeding up due to the liver enzymes involved becoming more active. And last but not least is side effects, which are in addition to therapeutic effects ranging from mild to severe and can stop patients from completing treatment, which also limits the usefulness of it, unfortunately. And therefore, minimizing side effects is a common goal in drug development and clinical practice. And next, I'll be talking about the similarities and differences in the structure of a drug-related molecule. And this is super important as it can impact a plethora of things, no matter how large or small. And things like functional groups, extra atoms attached to the main structure, its arrangement in 3D, and molecule size can greatly affect things like how a drug works in the body, how strong the drug is, how long the effects of a drug last, what side effects could occur, and how the body absorbs and processes a drug. So it's important to utilize these similarities and differences, as molecules with similar structures may mean similar biological targets and effects. And this speeds up drug development and provides a somewhat stable diving board into it as well. And this brings me on to my next point, which is modifying these molecules. And what is on the surface or exterior of a biological molecule determines its chemical behavior, and what is on the surface are functional groups. And chemical changes made to specific parts of a functional group will alter its properties and reactivity. And someone might modify a functional group to improve the pharmacokinetics or pharmacodynamics of drug candidates or enhance its biocapability. And when a compound with desired pharmacological effects is identified, chemists will introduce modifications to the chemical structure to create variations with improved properties, and these are called analogues. And these modifications will help improve the effectiveness, the delivery, and the safety of drugs. And an example of this is the improved targeting of drugs towards specific tissues and cells through the introduction of nanoparticle-based delivery systems. And this improves accumulation of drugs at the disease site and therefore increases the therapeutic efficacy and reduces off-target effects as well. And secondly, personalized therapies ensure tailored delivery systems to individuals based on age, genetics, and the state in which the disease is in. And this also optimizes efficacy and safety too. And an example of this is L-Dopa, which is an anti-Parkinson agent, and this was modified to improve bioavailability and efficacy, and this was achieved by introducing a hydrophilic group to it next I'll be talking about the classification of an isomer, specifically a type of isomer, and a crucial condition to classifying an isomer as either cis or trans is if there is not two non-identical groups attached to each carbon bond, then the isomer cannot be cis or trans. So after drawing the structure, meeting the condition, and extending the C to C double bond, we're able to see if an isomer is cis or trans, and cis is where the matching pair of substituents are on the same side of the dotted line, and trans is where the matching pair of substituents are on different sides of the dotted line. And next is geometrical isomerism and its relationship to drug molecules and their functions. And so first of all, what is geometrical isomerism? And it's a type of isomer whereby the molecules have the same structural formula, but the atoms making up the molecule are arranged differently in space. And due to the double covalent bonds in geometrical isomers, cis-trans isomers can exist in two forms. And now onto the significance, where geometrical isomerism has a significant effect on a drug molecule's functioning. And it can affect the pharmacological activity of a drug molecule, meaning the biochemical or physiological effects a drug produces. And an example of this is tranylcypromine and its cis counterpart, where it's shown here with these two diagrams. The cis is the top one and the trans is the bottom, and the trans is the one that we actually use. And this is because the trans one is an antidepressant due to its ability to prevent the breakdown of neurotransmitters in the brain, whereas the cis isomer is not as effective and is unable to prevent monoamine oxidase A from breaking down monoamine neurotransmitters in the brain. So we simply use the trans one because it's more effective than the cis one. And as well as this, it can also affect the chemical properties it has too. And an example of this is that trans isomers tend to be more nonpolar than cis isomers, as the charge is balanced on opposite sides of the double bond. And now chiral molecules, what are they? So they're molecules that do not match their mirror image and cannot be flipped or rotated to match. And it's most commonly occurring when a carbon atom is attached to 4 different substituents, and this is called a chiral center. And enantiomers are 2 versions of a chiral molecule that are mirror images to each other. And so if a mirror image of the molecule is identical, it is superimposable and therefore achiral, which means it is not a chiral molecule. And if the mirror image of the molecule is non-identical, it is non-superimposable and therefore chiral, meaning it's a chiral molecule. And this leads me to my next point, where optical isomerism is where each enantiomer causes plane polarized light to rotate 90 degrees in opposite directions. Enantiomers rotating the plane of polarization clockwise are known as the positive form, and enantiomers rotating the plane of polarization anti-clockwise are known as the negative form. And an example of how toxicity and metabolism are related is through thalidomide, where pregnant women were given the S enantiomer and R enantiomer versions of thalidomide to alleviate morning sickness, so a pregnancy symptom. And this unfortunately resulted in babies being born with severe birth defects such as limb deformities and damage to internal organs. And this is due to the R enantiomer metabolizing into the S enantiomer, and the S enantiomer is the one that causes the birth defects. So not only did we have the S enantiomer in there, we also had the R enantiomer metabolizing into the S. So the drug was essentially 100% S enantiomer and essentially 100% causing the birth defects. And as well as this, it also allows for personalized medicine, as people may respond differently to specific enantiomers. It's necessary to tailor drug therapies to the individual. And the following sections are regarding detecting drugs and designing and developing drugs. And there are 2 types of testing when it comes to detecting drugs: presumptive and confirmative. Presumptive is simply a quick preliminary tests to see whether there is the presence of drugs in the sample, whereas confirmatory tests are performed in a lab using more complex processes to provide a definite answer as to whether a drug is present in the sample. And due to these complex processes, it's also possible to find the quantity of the drugs as they're more specific and sensitive. And within chromatography, there is gas chromatography, liquid chromatography, and thin layer chromatography. Gas chromatography involves separating components in a mixture by vaporization and passing it through a column. And light chromatography, within spectroscopy, there is mass spectroscopy, infrared spectroscopy, UV visible spectroscopy, and Raman spectroscopy as well, of which mass spectroscopy is often combined with gas chromatography or liquid chromatography for enhanced identification, meaning it can detect trace amounts of drugs. And ultraviolet visible spectroscopy will measure the absorption of UV or visible light by a substance. And there are also techniques such as ion mobility spectrometry, and capillary electrophoresis. And the theory behind it is that all particles move at a specific velocity when under the influence of an electric field. The velocity at which particles move depend on their size, and charge, which allows the separation of the sample. And as the sample is entered into the tube on one end, on the other is a detector which reads the separated particles. And computers are also used in drug detection, design and development. So computer-aided drug design, or CAD, uses computational methods to identify, develop and optimize potential drug candidates. And there are various ways in which CAD aids drug design, for example, molecular modeling, virtual screening, quantitative structure-activity relationship or QSAR, pharmacokinetics and toxicity prediction and quantum computing. And I'll talk about two of these in greater detail. So pharmacokinetics and toxicity prediction focuses on ADMET, how a drug will behave in the body regarding the following points: absorption, A; distribution, D; metabolism, M; excretion, E; and toxicity, T. And it's a cost-effective and efficient way as time can be focused onto molecules and compounds that do show promise. And it's also a safer starting point, as any molecule or compound that could be deemed too toxic or cause undesirable effects are identified and disregarded as potential candidates. Quantum computing optimizes processes by analyzing protein hydration using algorithms to precisely place water molecules in protein packets, what drug development is dependent on, and predicting how small molecules interact with larger biological targets. And quantum computing has the potential to streamline submissions to regulatory bodies, as it would provide more precise rates of error and also simulation. And regarding AI is assisting greatly in drug design, detection and identifying new drug molecules, which is something I'll touch upon a bit later. But one thing I wanted to mention is that it's also identifying new ways in which existing drugs can help in the treatment of other conditions. As technology becomes smarter, it's also imperative to use it to not only help new developments and conditions, but ones we might have previously thought we had found solutions for. And so there are many ways in which to identify new drug molecules. However, I'll be discussing three of these in greater detail: machine learning and AI, high throughput screening, and ligand-based drug design, or LBDD. And deep learning models use neural networks to predict molecule properties such as absorption, distribution, metabolisms, and safety, etc. And there are also neural networks with transfer learning, where it can also store knowledge from one task and transfer its learning to another problem, which is kind of what we talked about, what I briefly touched upon in AI. And this also improves the predictive performance of drugs at later stages as well. High throughput screening accelerates target analysis as a large number of molecules are tested in a cost-effective manner, and molecules that meet the biological target in the desired way are called hits or leads, which are suggestions for starting points for further study. And in ligand-based drug design, or LBDD, researchers build upon existing data and attempt to predict and identify patterns in ligand activity and use them to assist in designing new molecules that are more effective and selective. And then that's the end of my presentation. Thank you for listening. I hope it was informative and have a nice day.